Welcome, I'm Matthew Solomon, and on this video, we're going to be talking about GMOs, genetically modified organisms. Genetic engineers like the ones behind me work day and night to further the development of gene and DNA modification. But to further understand GMOs, we must first understand DNA, genes, and proteins. Here today to talk about the structure and role of DNA and the function of genes and proteins is the famous Dr. Know-It-All from Harvard University. Hi, I'm Dr. Know-It-All here at Harvard University. I'm here today to talk about DNA, genes, and protein. Let's take a look at a strand of DNA. DNA, as shown in this picture, has a linear form, and each molecule of DNA is a double helix formed by nucleotides held together by hydrogen bonds between GC and AT. As shown in this DNA strand, nucleotides use sugar as shown in blue and phosphates as shown in red, and the four bases adenine, thymines, cytosines, and guanines to form the DNA strand. DNA, or deoxyribonucleic acid, is the self-replicating genetic information found in all living organisms. DNA provides the instructions for the genetic traits, and now it is very important that you get the right strand of DNA so you don't get an insertion or deletion that can cause mutations. Now let's talk about the function of genes and proteins. Genes are segments of DNA that contain hereditary information. DNA encodes protein, and protein replicates to maintain DNA. Let's understand more about the history of DNA. Matthew, can you help us out with that? Thank you, Dr. Know-It-All. DNA was discovered in 1866 by Gregor Mendel while he was studying pea plants. For more than 50 years, no significant studies were made. In 1953, James Watson, Francis Crick, and Rosalind Franklin discovered the structure of DNA. They used X-ray crystallography to find out the structure of DNA, which is now called today the double helix. In 1962, a Nobel Prize was awarded for their research. Now let's talk about genetic engineering and how it relates to GMOs. Genetic engineering is the modification of an organism's characteristics by manipulating its genes. Let's take a look at the process that these genetic engineers take to make a GMO. Corn is a very common GMO. It has been genetically modified to resist insects but would otherwise kill the corn. This is how we create the GMO for corn. Step 1. DNA extraction. Extract bacteria from soil, which is resistant to insects. Step 2. Gene cloning. Step 3. Gene design. Step 4. Transformation. And Step 5. Back cross breeding. It takes 6 to 15 years for a GMO to be fully developed. Now let's try to have some fun and try to create a whole new GMO. Let's pick our desired trait. Since this is my video, I chose a trait that can survive with little to no water. We're going to take the desired traits from a cactus that can survive with little to no water and put it into a delicious, juicy tangerine fruit. Our new GMO will be called the cactarine. This is how we'll create the cactarine's GMO. Step 1. Extract the desired trait from the cactus. Step 2. Cloning and designing the genes. Step 3. Transformation. Step 4. Plant breeding. And finally, you have the cactarine. These cactarines are sure delicious. The benefits of this GMO is it can almost be produced anywhere, including starving third world countries. Potentially, this cactarine can change the world. One of the risks of this GMO is it can cause new food allergies. What other things do we need to consider with our GMO? What stakeholders can be affected with this GMO? Since this GMO can be produced almost anywhere, U.S. farmers and exporters may be negatively impacted. Whenever we're talking about GMOs, there is always a moral and ethical consideration. Some people disagree of scientists taking over for Mother Nature. But for me, all the benefits for the cacturine outweigh all the risks. This concludes our episode of GMOs. I hope you enjoyed it as much as I did. And hey, 5 to 16 years from now, don't be surprised if you see the cacturine.